Welcome everyone to the wonderful world of protein synthesis. So in this section, like a lot of our other sections, there's a fair amount of vocab. So we came up with some analogies that hopefully will help you kind of solidify these ideas, but it's something that we're going to say over and over again because it's not everyday language. So just kind of keep that in mind, focus on the words, and we'll kind of do some things in class to reinforce it. But we're making proteins. That's all what we're talking about today in terms of how that coded message in DNA actually is expressed in your body, and that's through proteins. So as we go through, we want to be able to talk about the different types of RNA, and we're going to be able to compare and contrast this process called transcription and translation. So here we go. So DNA and RNA have some similarities for sure. They're both something that we refer to as nucleic acids, but there are some definite differences. So when we talk about RNA, it's still long chains of nucleotides. Okay, it's still nucleotides because they are nucleic acids. Okay, RNA is a nucleic acid, so it's made up of nucleotides just like DNA is. So DNA stands for deoxyribonucleic acid, where RNA stands for ribonucleic acid. So some differences and that's exactly what I'm saying right here. The sugar is ribose, so this is our sugar for RNA, compared to deoxyribose is the sugar for DNA. And you could say, boy, that's pretty similar. The only difference is whether it's an H or an OH. So yeah, they are very similar. It's a sugar, but DNA deoxyribose, whereas RNA is ribose. But it is a single strand. So if you notice that RNA is a single strand where DNA is a double strand, it has both sides, 1, 2, where over here it's just got one side. So it's single stranded. And instead of there being thymine, we have a different nitrogenous base or nitrogen containing base called uracil. So the idea is it's still Chargaff's rule, and Chargaff's rule said that for every C there's a G, and for every A, there's a T. Well, that's true with DNA. However, when we get to, it's not going to show up. When we get to RNA, and for every A now there's going to be a U. So we replace the T with a U. So still kind of the same idea of Chargaff, except for now we got to substitute a U instead of that T. But when we talk about RNA, it's considered like a disposable copy. So we make a copy, we use it, and then we dispose of it. We don't keep it. The DNA is the master blueprint that is very sacred, and we don't want to screw up, so we don't want to take it to the job site and have it potentially get wrecked. So there's 3 different types of RNA, but the main job of RNA is to make proteins, which we refer to as protein synthesis. Synthesis is to put together, to make, so we're making proteins. Proteins do most of the work in the cells and are required for the structure, function, and regulation of body, tissues, and organs. So there's, again, 3 types. There's mRNA, there's rRNA, and there's tRNA. And we'll talk about the differences of those 3 types of RNA as we kind of move forward. Okay, so there's 3 types, though: mRNA, rRNA, and tRNA. The 2 that we're going to be most concerned about are the mRNA and the tRNA, where rRNA is more the site, the place where it occurs. So mRNA. So when we talk about mRNA, it stands for messenger RNA. And what it does is it takes a copy of the DNA from the nucleus and it takes it out to the cytoplasm. It carries the message that the DNA had. Messenger RNA makes a copy of the DNA and it carries it to the cytoplasm, specifically the ribosomes in the cytoplasm. Again, rRNA is said more of a location. It's what makes up the ribosomes. So rRNA stands for ribosomal RNA, which is what we talk about when we talk about ribosomes. And ribosomes are where proteins are made. That's where protein synthesis is going to occur. That's the site that we're making proteins that are floating in either in the cytoplasm or attached to the endoplasmic reticulum, and they're the rough bumps on it in the rough endoplasmic reticulum that we talked about back in the fall. And then the last type of RNA is something called tRNA, or transfer RNA. And what it does is it transfers amino acids to the coded message, okay? And then this decodes the message into a protein. So tRNA is this kind of clover-shaped thing, where mRNA is a single chain, single strand, linear, and rRNA is kind of the chunks, it's the pieces, it's the place, it's the site that make up the ribosomes. So how does that DNA message go from DNA to protein? Well, there's two steps. It's called transcription and translation. So when we talk about transcription, we are transcribing something from one form of nucleic acid to another form of nucleic acid. We're going from DNA to to mRNA. And again, we need enzymes, type of protein. We need enzymes that help do this. So a lot like DNA polymerase and DNA helicase, there's an enzyme called RNA polymerase, which is used to separate the DNA into two strands. So this should look pretty similar to DNA replication. And then we're going to bring in the complementary base pairs, pretty similar to DNA replication. However, it's a different type of enzyme, so therefore it's bringing in different types of nucleotides. It's bringing in the mRNA nucleotides instead of the DNA nucleotides. So in order to do that, it separates the two strands and it uses it as a template to copy it into mRNA. Remember, complementary base pairing, we use a single side to build a complement using Chargaff's rule. And then RNA polymerase must find the promoter region, which is where it will start copying the specific sequence of DNA. So DNA is really, really strong, and we don't just code everything. We need to give a start signal, and that's this idea of a promoter, and it tells the DNA where to start coding for that protein. And this all takes place in the nucleus. So transcription takes place in the nucleus, but it goes from DNA to mRNA, and this occurs in the nucleus. That's kind of the big thing there. Complementary base pairing, A's go with a U, and C's go with a G. Okay? And a T goes with an A, and a G goes with a C. So C's and G's are the same. The big difference here is an A's go with U. That's really the big kind of difference here. So an analogy to kind of help you hopefully remember transcription is think of the transcription as a process of creating a copy of a page in a book. So let's say you are at the library and you find a really good recipe for chocolate chip cookies. But for some reason, the librarian is saying you can't take that book out of the library. It's special. You can look at it, you can view it, but you can't remove it from the library. It's on like reserves and you can't take it out. So the idea is, how would you make those cookies at home? Well, you'd have to make a copy. So in order to do this, you take that book, you go to the copy machine, and you make a copy. So in the nucleus, we have in the library, which is the nucleus, we have a book that has a particular message that tells us how to put things together, make chocolate chip cookies in this case. So what we do is through the process of transcription, we take that page from the book and we make a copy, and then we can take that copy out of the library with us. Similar in DNA and RNA, the DNA is the book. The DNA is the book that can't leave the library, and the copy of the recipe is the mRNA. It's the mRNA. Okay, so in order to make that copy, you have to go through this process called transcription. Okay, so the nucleus is like the library, and that copy is called mRNA. So putting all these things together, we have to kind of, this says remember, we actually didn't do this section this fall, but amino acids are the building blocks of proteins, okay? It's the building blocks of proteins. It's the monomers of protein. It's the single unit that we put together to make a protein. So it's these long chains called polypeptides, and when we put a bunch of different amino acids, so we have a bunch of amino acids, when we put them together in a long chain, we get this thing called a peptide, and then when we fold them in this perfect sequence, we get something called a protein. There are 20 different amino acids that exist that, depending upon the combination we put them, the order in which we put them in this peptide bond, depends upon how it's going to fold, and depending on how it folds, depends upon what protein we're going to produce. So think of the amino acids as letters in the alphabet. And if we have 20 different amino acids, it's kind of like we have 26 different letters in our alphabet. And those 26 different letters in our alphabet can be put together in different sequences to make different words, just like these 20 amino acids can be put together in order to make different proteins. So in order for us to know what amino acid is going to be brought in, it has to do with the sequence of the nucleotides, which is coded for in the DNA. So that DNA is in the nucleus. It can't go to the ribosomes because it's in the nucleus, and that DNA is never going to leave the nucleus except for during mitosis when the nucleus dissolves, but it never leaves the nucleus. So we made that copy of that chocolate chip cookie recipe to take home with us, just like we made a copy of that DNA in the form of RNA, which we're going to take to the ribosomes with us. So mRNA has a sequence of nucleotides that will code for a specific amino acid. Okay, the sequence of the nucleotides code for specific amino acids. And when they look at which amino acids, it reads it 3 letters at a time. And these 3 letters on mRNA are called a codon. And the idea is if you go U, C, G, if you start in the middle with this one, you go U, and then you go C, and then you go G, this is going to code for the amino acid of serine. So if we start here and go C, A, G, then we're going to say, hey, that must be, oops, I coded that one wrong. C, A, C, then it's going to be histine, and then lastly, G, G, U would be glycine. And don't worry, you are not expected to memorize this chart. You will get a codon chart. Now, this particular chart codes off of mRNA, but there are other charts that might code off of DNA or might code off of tRNA. It just kind of depends upon the chart. So you just kind of make sure that you take note of that this particular one is an mRNA. So again, codon is a 3-letter combination, but we need to have a start codon. In order for us in the ribosomes to say, where do we start coding for that polypeptide, we need a start codon. And on mRNA sequence, that's an AUG. We need to have an AUG. We have so much DNA that we don't just start coding everywhere. We need a start codon. And then we look at 3 letters at a time, and we keep tagging on new amino acids until we hit a stop codon. So if you look, here's a start, a stop, here's a stop. And we keep going on and on and on until we hit one of those. So we just keep adding amino acids, amino acid, amino acid, amino acid until we hit the stop codon. Okay, and that tells us to stop putting together amino acids, which is then saying we're done making that particular protein. And that process of what we're bringing in these amino acids, that's a process called translation. So when we go through the sequence of nucleotides, mRNA serves as instructions for which amino acids will be joined together to form a protein. So we have a particular strand of mRNA that we are going to do complementary base pairing, Chargaff's rule, once again, and it's going to bring in and it's going to look at 3 letters and only 3 letters at a time. And what's the complement of a C? It's a G. What's the complement of a G? It's a C. And what's the complement of G? It's a C. Okay, for every A, there's a U. For every A, there's a U. For every G, there's a C. The only difference between Chargaff with DNA is for every A, there's a U. Otherwise, C's goes with G's. But keep in mind that a U is also going to go with an A. So when we go mRNA to or RNA to RNA, we're using U's instead of T's. And where this occurs is in the cytoplasm, specifically on a ribosome. Okay, so that's the structure that protein synthesis occurs. And we're looking at complementary base pairing. But these 3 tRNAs bring in a specific amino acid. These 3 tRNAs bring in a specific amino acid. These 3 bring in a specific amino acid. And that's what this chart shows us, and that's what this chart. So whatever 3-letter sequence we have gives us a specific amino acid, and 3 different letters is going to give us a different amino acid. Okay, that's translation. So steps of translation. The mRNA attaches to a ribosome. So again, this occurs on a ribosome. Protein synthesis occurs on ribosomes. That's the function of a ribosome, is to make proteins. We look at 3 nucleotides at a time, and then we need a start codon. And then each codon codes for an amino acid. So if these are called codons, the complement is called an anticodon. The opposite of a codon is an anticodon. So the mRNA single-strand on the ribosome codes for 3, and then the complement is going to come in, and that brings in a specific amino acid. And depending upon what anticodon we have, depends upon what amino acid that was once attached here. But when this comes through and we form an amino acid, the amino acid was here, it is now attached right there, then this goes away and it disappears. And now we have a 2 amino acid sequence. So then the next codon comes along when it gets the complementary anticodon that brings in a specific amino acid, and then what's going to happen is we're going to form a bond here, and then this is going to go away, and then the next one comes in, and then this is going to form here, and then they go on and on and on. They keep building an amino acid chain. Each amino acid is brought in to create a polypeptide chain, which eventually will fold and make a protein. Okay, so again, if we go back to our library translation of that chocolate chip cookie recipe, transcription occurred in the library, in the nucleus. We have the sacred special library recipe book that has instructions on how to assemble a cookie, chocolate chip cookie, right? And that's DNA. But you can't take that book home, so what you do is you make a copy of the instructions, the recipe, the DNA, in the form as the form of mRNA, and then that copy can then leave the library. That's transcription. Now what you do once this copy of that recipe is at home, you can take that recipe and you can follow the instructions that will help you assemble and ultimately make the cookie. And in this case, the cookie is the protein that we're referring to. Okay, the cookies are the proteins. So if we kind of look at our analogy, when you go home, where are you going to assemble the cookies? That's at home. That's like where the proteins are made, on the ribosomes. The copy of the recipe still is our mRNA. The ingredients of our cookie are the amino acids. So we need flour, sugar, butter, whatever. Those are the building blocks of a cookie, just like the amino acids are the ingredients or the building blocks to make our protein, which is our final product in our recipe. Is we're going to make these delicious chocolate chip cookies. And all of this takes place, this process takes place on the ribosomes. Okay, so that is protein synthesis. There's a lot there. We will do our best to keep reinforcing these ideas in class. If you have questions, please